First of all, I would like to thank the organizers for including our work in the program of this meeting and for a chance to visit Russia and visit Moscow. Большое спасибо. Today we'll be discussing briefly some relationships between hormones and aging. Is this thing working, or do I have to say next? Okay. It's very well documented that there are major changes in the production and release of hormones with aging. And this is well documented and clear for many decades now. What is maybe less clear is how the functional meaning of these changes should be interpreted. We can look at these changes as symptoms of aging, that, for example, just as the function of muscular system or function of the lung declines with age, so does the function of endocrine glands. It may also be looked at as one of the causes of aging, that some of the mostly unwelcome changes that happen to us as we get older reflects lack of hormonal stimulation and reduced action of these various hormones on their targets. However, the same changes can also be looked at as perhaps naturally evolved protective mechanisms. So, for example, we can think of age-related decline in growth hormone release as a cause for declining muscle mass and increased accumulation of adipose tissue. But we can also look at it as a mechanism that reduces the chances that we'll get cancer or diabetes. And in addition to these relationships between age-related changes in hormonal system and aging, whatever they may functionally mean, there is also another relationship which appears counterintuitive, but thanks to research of last 15, 20 years, I think is by now well documented. Namely, that major defects, major decreases in hormone action can lead to extension of longevity. And what this implies is that normal actions of these hormones, perfectly normal actions of normal physiological levels of these hormones, are apparently not optimal for life expectancy, because if you reduce these signals, the life can be extended. So this is this aspect of interaction of hormones and aging that we are working with. Next. Next, please. And all of this has started with a demonstration by Dr. Holly Brown-Borg, who at this time was working in our laboratory, and she's here in the audience, by the way, showing that a particular mutant, shown here in green, lives much longer than normal siblings of the same mutant. And as you can see, there was a major extension of both median and maximal longevity, suggesting that the process of aging is slower in this animal, so perhaps delayed would be a more accurate term. Now, what is this mutant? Next. This mutant was discovered. It was a spontaneous mutation in the research mouse colony at Iowa State University in Ames, Iowa, which was discovered in late 50s. And because this institution is located in Ames, Iowa, they call it the Ames dwarf mouse. And these animals have a mutation of a gene which is involved in development and differentiation of anterior pituitary. And when function of this gene is lost, 3 hormone-producing cell types in the pituitary do not develop. These are the cells producing growth hormone, prolactin, and TSH, So the animal ends up not having these three hormones. And by the way, we humans have the same gene and possibility of the same mutation. And there are reports in medical literature of people with mutation of the same gene, which is called the prophet of pituitary factor 1, PROP1. And these individuals are dwarfed and hypothyroid, just like Ames dwarf mice. Next, please. Now, to show you some data from these animals, without growth hormone producing cells, of course, they produce no growth hormone, so there's no growth hormone in the circulation. Without growth hormone, the production of IGF-1 by the liver is drastically reduced. Circulating levels of IGF-1 are very low. For this particular study, we used radioimmunoassay, and in this radioimmunoassay, the circulating IGF-1 levels in the Ames dwarf mouse were in fact below the limit of detectability. Without growth hormone and IGF-1, not surprisingly, they are very small. You can see in the top line, they are only about 1/3 size of the normals at this age when they were looked at. And they have reduced levels of insulin and reduced levels of glucose. And this concomitant presence of these two changes implies that they are more insulin sensitive. And I think after Professor Anisimov's lecture, you are hopefully sensitized to the importance of insulin in the control of aging. And I will be returning to this point several times during my talk. Next, please. Another animal that we are using for our research is an animal with targeted deletion or knockout of a gene coding for growth hormone receptor. So this animal has no growth hormone receptor and is, as a consequence, growth hormone resistant. So it produces growth hormone. In fact, it produces more growth hormone than the normal mouse because the negative feedback doesn't work. But without a receptor, it doesn't respond to it. It's small and has functional deficiency of growth hormone. This endocrine syndrome, by the way, mimics almost exactly the symptom of Laron dwarfism in the human. Next, please. These animals are also long-lived. The mutants are green on the right, normals red on the left. Next, please. And in our colony of these animals were developed in the laboratory of Dr. Kopczyk at Ohio University in Athens. That's Athens, Ohio, not Athens, Greece. And in our colony of these animals, we had one animal that missed its 5th birthday by a week, which is something mice normally don't even approach. So as far as we know, he was the oldest mouse on record. But of course, that's the case of one, so we don't get too excited about it. But he was just an interesting outlier. Next, please. Now, what do these animals die from? These are data from pathology and cause of death deduced from histopathological finding at the natural end of life. And we are comparing Ames dwarfs on the right with normals on the left. And there is a suggestive decrease in the incidence of cancer. And these animals lived on the average 3 years rather than on the average 2 years as the normal animals. So they lived another year and yet had less cancer. For particular types of cancer, in this case adenocarcinomas, there was in fact a significant reduction in incidence. And there was a significant increase in the proportion of animals that didn't have any obvious histopathological findings explaining death, so probably died from some functional problems which did not have a morphological expression, perhaps cardiac function. Next, please. Very similar data for this Laron dwarf mice or growth hormone receptor knockouts. Here there's a significant decrease in overall incidence of cancer. Again, significant protection from adenocarcinomas and significant increase of these animals that don't have gross histopathological changes at the end of life. Next, please. One of the characteristics of these animals that attracted our attention was that in addition to living long, they look young. And in fact, in many physiological characteristics that we can measure, they appear to be biologically younger than their chronological age would suggest. And the data that we think are maybe most interesting, especially if you think of possible translation of this type of findings to the human, is that they seem to maintain their cognitive function into advanced age. So what this image shows compares normal animals to these long-living mutants. And in each case, young adults and old animals, which in this case were approximately 2 years old. And you can see first that these mutants on the test, this is a passive avoidance test, and the height of the bar, of this dark bar, indicates how well the animals learn and remember. So first of all, we see that these animals are on this particular test performing as well as the normal animals when they are young. And at the age when the normal animals have the expected decline in memory, learning and memory, the mutants do not. And next, please, is very similar data for the Ames dwarf, a significant age-related decline in cognitive function in the normal, and no significant change, just a small non-significant trend in the mutant. Next, please. So in overall, these animals have what sometimes is referred to as the longer health span. In other words, they remain in the physiological functional state of being young and healthy longer. And the list of these changes comes from work in several laboratories and also on another dwarfing mutant, which has the same endocrine defects as the Ames dwarf mouse, called the Snell dwarf mouse. And collectively, this work indicates that there is a delayed loss of cognitive function, delayed development of cancer, delayed loss of immune system. This was estimated by the ratios of memory and naive T cells. There is also delayed collagen aging and delayed cartilage aging and osteoarthritis development. And Dr. Arum, working in our laboratory, added recently to this list that they also have delayed loss of physical strength and delayed loss of the sense of balance. So the changes which associate with human frailty and falls and other significant issues for the elderly, these animals seem to be protected from this. So they not only live longer, but they stay in good shape longer. Next, please. So why do we want to work with these animals? Well, one advantage is that their longevity, as Dr. Rees was explaining before lunch, is traceable to a known mutation. So we know that there is a gene which we know what it does, and whatever is different about these animals is caused by this single mutation. So this makes it a little easier to look for possible connections and mechanisms. Another advantage of these animals is that since this is a genetic condition, we know right from the beginning which one is going to be long-lived and which one is going to be normal. So we don't have to wait for them to become old to know who is long-lived and then worry about age-related changes. But we can look at young, healthy animals and differentiate between those that are long-lived and those that are not. Next, please. So and next. There are some arrows there that should appear. So anyway, what this data imply that if you're a mouse, and if you are genetically deficient or genetically growth hormone deficient or genetically growth hormone resistant, you live longer and you have various symptoms, next please, of delayed aging. And of course, we could not prove from this data, even though we believe that this increased longevity is largely expression of delayed aging, we couldn't conclusively prove it because there is also possibility of reducing age-related disease and so forth. But the fact is that animals with these endocrine defects live longer. Next, please. So when we talk about growth hormone, we of course have to think about IGF-1. And what this diagram shows, and I'm sorry that, that I don't have a pointer, but I don't think I do. No. But you can see on the top of the slide that the pituitary releases growth hormone. It acts on the liver, causes production of IGF-1, insulin-like growth factor 1, which acts as a hormone. Endocrine IGF-1 circulates and affects many targets. There is also local production of IGF-1 in different organs, some of which is regulated by growth hormone and some is not. And this is an important difference. And in turn, action of IGF-1 is modulated by IGF-binding proteins, IGF-BPs. There's at least 6 of those in mammals. And their levels, in turn, are controlled by specific enzymes, proteases, which degrade them and determine the level of free biologically available IGF-1. So this is roughly the interaction of this system, which is sometimes called the somatotropic axis. Next, please. Here, work in various laboratories resulted by now in identification of a rather long list of mutants that are long-lived, and they have defects at various phases of growth hormone signaling, IGF-1 signaling, or IGF-1-related downstream mediators. And so this certainly confirms the involvement of somatotropic axis in the regulation of longevity. In general, the effects of reducing growth hormone signaling have greater impact on longevity than the effects of IGF-1 signaling. There are many possible reasons for this. The reason that one of the reasons which interests us most is that it may indicate that growth hormone affects aging by mechanisms other than reducing IGF-1. And we think that that's exactly the case, that growth hormone modulates sensitivity to insulin and modulates secretory function of adipocytes. And these are not IGF-1-mediated effects, but they are important for longevity. Next. So with this information, of course, we would like to know what are the mechanisms, what connects these endocrine defects with longer life. Next. And the data available to date, and this is a result of work in a number of laboratories, there's a whole list of candidate mechanisms, and I have arbitrarily grouped them here in 4 groups. Mechanisms related to growth, which include, of course, IGF-1 and growth hormone itself, and also mammalian target of rapamycin that you heard about from Professor Anisimov's lecture. And I think you will hear more about it in the course of the meeting. There are also defense mechanisms. Work of Dr. Brown-Borg was important here in elucidating those. And stress resistance, Dr. Richard Miller made major contributions here. And there are also changes in inflammation, namely decline in pro-inflammatory and the increase in anti-inflammatory factors, and I will return to this briefly later on. And there are various metabolic adjustments, and this includes fatty acid oxidation, changes in RQ that Dr. Rees mentioned, and the metabolic adjustments which we are particularly interested in is enhanced insulin sensitivity. Next, please. I already told you that these long-living mutants have lower insulin and lower glucose. Here it's shown for both Ames dwarf and growth hormone receptor knockout. Next. And in the next, please. And in the Ames dwarf, if we compare Ames dwarfs in yellow to normal animals in red, we can see that they are more insulin sensitive. These are changes in glucose after injecting the same dose of insulin per gram body weight. And if we inject these dwarf mice with growth hormone for a few weeks before doing this kind of experiment, then they lose this insulin sensitivity, and we get the green curve, which is not different from genetically normal animals. Next, please. Now, some of our evidence for this coupling of what happens to insulin sensitivity and what happens to longevity came from the studies where we looked at interaction of these longevity genes or these mutations that cause life extension with calorie restriction. We used 30% calorie restriction. We forced the animals to eat about 30% less than they would eat if they had unlimited access to food. Started this at about the age of 2 months and continued for the rest of their life. And if we did this with Ames dwarf mice and their normal siblings, they both lived longer. Both the normals lived as long as dwarfs that eat all they want, and the dwarfs on calorie restriction lived even longer, as shown on the graph on the left. When you look on the right, there is essentially a mirror image of these changes in insulin sensitivity measured by this insulin tolerance test. Normal animals in red, when you put them on calorie restriction, they become more sensitive, green. Dwarfs are very sensitive to begin with, the blue line, and if you put them on calorie restriction, they are even more sensitive, yellow line. When we wanted to repeat this experiment next, please, in the growth hormone receptor knockouts, we got a different picture. Without going into details, the mutants, the normal animals lived longer on calorie restriction, as you would expect, but the mutants did not. There was a small difference for females, no difference for males. Next. And essentially, the same thing was true for insulin sensitivity. In the top 2 panels, when we put normal animals, either gender, on calorie restriction, they become more insulin sensitive, but the same calorie restriction regimen had no effect on insulin sensitivity of these mutants. So whatever happens or does not happen to insulin signaling also happens or does not happen in terms of longevity. Next, please. So because of this, we, of course, became interested in insulin signaling, and lots of work that we've been doing for last now over 10 years, and studies that we are doing now concerns looking at specific mediators of insulin action in different insulin target organs, particularly in the liver and the skeletal muscle. Some of those are shown here. And the addition to this work, which we are very excited about, is that Dr. Kopczyk, our collaborator, recently generated animals in which growth hormone receptor is deleted specifically only in one tissue. So there are 3 new lines of these animals. There are animals with growth hormone deletion in the muscle, in the liver, or in the adipose tissue. And we are just beginning to look at characteristics of insulin signaling in these animals. And since I was told that this lecture was supposed to be not overly technical, I will not try to summarize this work or show you this data, but simply indicate that this type of work is pinpointing the molecular differences in individual tissues which explain changes in insulin signaling at the whole animal level. Next, please. One aspect of these animals that I would like to discuss with you today is how these metabolic changes, and particularly enhanced insulin signaling, relates to the body composition of these animals. As I already mentioned, these animals are long-lived. They stay healthy longer than normal animals. They are hypoinsulinemic, they have low insulin, and they are very insulin sensitive. So these are phenotypic characteristics of someone who is healthy, lean, and fit. These are the kind of characteristics you can promote by calorie restriction, by dieting, by exercise. However, these animals are not lean. In fact, they are obese. These data are generated by Dr. Berryman. We have similar data for our animals, but hers are more detailed, so I'm showing her data. On the top is the absolute fat content in these mutants, but as you recall, these mutants are much smaller So I think a more meaningful comparison is looking at the amount of fat as the percentage of body weight. And that's the lower two panels. And you can see that these animals have a lot more fat than their normal siblings. The mutants are in open symbols on the top of this graph. And the normals are with full symbols on the bottom. So you see that males have almost twice as much fat as normals, and females have about 50% more. So we decided to make some predictions from this body composition. If they have these favorable metabolic characteristics and increased amount of fat, this must mean that this fat, for some reason, is less detrimental. It does not have the impact on metabolic pictures, on glucose homeostasis and so forth, as we would normally expect. And to try to probe this suspected difference, we decided to remove surgically some of the fat depots and see what will happen. And the rationale for this study is summarized on the next slide, please. It was already shown in the laboratory of Dr. Barzilai, who also is here in the audience, that in the rats, if you removed intra-abdominal fat, they become more insulin sensitive. They become apparently healthier because they, in fact, live longer. So there's a very dramatic positive effect, beneficial effect of fat removal. Now, we already knew that in these animals, there is excess of fat, and yet they have this metabolically healthy profile. So we thought that this fat apparently is less detrimental in these animals. So if we remove it, we expect it to get less benefit, or perhaps to get no benefit. So next, please, what we did, we removed most of the fat around the epididymis in male mice and most of the fat around the kidney. And this represents majority of intra-abdominal fat in these rodents. And then we looked at various characteristics, expression of genes in different tissues and so forth and so on. And the data that I want to show you start with the next slide. Here are levels of insulin and glucose. And in each case, going from the left to the right, you are looking at the data from normal mouse that had sham surgery, normal animal that had these fat depots removed, a dwarf that had a sham surgery, and the dwarf that had the fat removed. So you can see that this fat removal reduced insulin in the normal animals, as expected from Dr. Barzilai's lab in the rat and from some clinical studies with removal of mental fat. But this did not happen in the dwarfs, where the insulin was low already, but it didn't become any lower. Insulin didn't change in the normals, and it went up in the mutants, suggesting that the change in insulin sensitivity would be, if anything, opposite. So we tested this directly, next slide, by looking at insulin tolerance, top panel, and glucose tolerance, lower panel. Sorry, back, please. And if you can see on the top that the animals, these mutants shown in green, are more sensitive to insulin, more sensitive to insulin than the normals shown in red. Big difference, as we have seen in previous studies. When we remove the visceral fat, the normals become more sensitive. You see the line is lower. But the mutants become less sensitive. There's a change in opposite direction. And if we test them for glucose tolerance or ability to clear injected glucose from blood, we get essentially the same picture, that their ability to handle glucose improves by removing fat in the normal, but it deteriorates in these animals. So apparently this fat is doing something else. Rather than promoting insulin resistance, it appears to be promoting insulin sensitivity. Next slide shows one possible reason for that. If we measure adiponectin, it's a product of adipose tissue that, in addition to anti-inflammatory and anti-atherogenic effects, also promotes insulin sensitivity. And what this image shows is that, first of all, if you compare the white with the white bar with the red bar, solid white with the solid red, you see that adiponectin levels in these mutants are much higher than in the normal animals. And we have seen this, and it was also seen in the Kopczyk's lab. It's a consistent finding. If we remove these fat depots from the normal, nothing happens to adiponectin levels. So most of adiponectin in these normal mice must be coming from subcutaneous fat, not from visceral fat. But if we remove visceral fat from these dwarfs, adiponectin is normalized, goes down to the level we see in wild-type animals. So apparently, in these animals, visceral fat is the major source of adiponectin, and this probably is one of the reasons why they are more insulin sensitive. Since then, we also looked at pro-inflammatory cytokines, IL-6 and TNF-alpha, and production of these cytokines by intra-abdominal fat is, in fact, lower in these long-living mutants than in the normal animal. Next, please. So now for almost half an hour, you were patiently listening to me talking about dwarf mice. And I hope that you agree with me that they are interesting. They're also absolutely adorable. But you may ask, what's the relevance of these mutants with really very extreme phenotype to anything else? Well, to try to put it in the sort of broader picture, next slide, I wanted to bring up the issue which was already brought up this morning, namely the homology of the genes which are involved. In 1997, which was exactly 1 year after Holly-Brown-Borg reported that Ames dwarfs are long-lived, laboratory of Dr. Rufkun reported that the key longevity gene in C. elegans, called DAF2, is homologous to genes coding for mammalian insulin receptor and mammalian IGF-1 receptor. And you will recall that these long-living mutants have essentially no circulating IGF-1, and they have big shift in insulin action. They release relatively little insulin, but they are very insulin sensitive. So you can see the parallelism and how nicely this data fit. And this homology of the genes involved in controlling aging in invertebrates and in mice caused many people to look at other genes, and by now I think we have considerable evidence. Next, this is summarized here in the review article by Dr. Longo, who will speak at this meeting also, and Dr. Finch, showing these homologies which in fact extend from yeast through C. elegans and Drosophila to mice and also to humans. And without going into details, in each of these species there is evidence that signaling by insulin IGF-1 or homologous molecules in invertebrates is importantly involved in the controlling lifespan. And in general, if you turn down this insulin signaling, you extend longevity and delay aging. Next. Now, there are also indications that some of these findings in mice, which I was discussing, apply to the humans. And there are important species differences. For example, the major cause of death in laboratory mice is cancer. In human, it's slightly more common to die of cardiovascular causes than on cancer, even nowadays with considerable improvement in cardiovascular disease prevention and treatment. So the causes of death are not the same. There are other differences. I think we are biologically a long-lived species, so perhaps manipulating human lifespan is harder than manipulating lifespan of a short-lived species. But there are also some rather striking parallelisms. And some of them are shown here. These dwarf mice, as I was emphasizing, have low insulin, low glucose. They are very insulin sensitive. They have high adiponectin. And there is evidence that people with extreme longevity have very much the same physiological characteristics, that in many studies, centenarians were found to have these blood parameters resembling what we find in long-living mouse mutants. Now, next, please. I think the strongest evidence for similar mechanisms controlling longevity in people and in these mutant mice come from various studies related to insulin signaling and insulin resistance. You already heard from Professor Anisimov about effects of anti-diabetic drugs of longevity. This here summarizes some work from Leiden University, from a Leiden longevity study, and at least one member of this group is going to speak at this meeting, Dr. Lynn Slagbaum. I don't know if she's already here. In this study, what they did, they identified long-lived families, which were identified as families where at least 2 siblings lived to be 90 years old or older. And then they looked at children of these people. So these were people who were about 60, and compared them to their spouses or partners. So this minimized the effects of environment, social, economical circumstances, diet, and whatnot. Now, I should emphasize that these comparisons were done on healthy people, okay? Offsprings of these long-living families have lower incidence of diabetes, lower incidence of elevated blood lipids, lower incidence of elevated blood pressure. They also have lower mortality. So they are significantly healthier. So if they took these 2 groups without selection, the differences in all these parameters would be much greater. But for these particular studies, they eliminated people with diabetes. They look only at people who are clinically normal and compared various parameters of insulin signaling in these folks. And they found that progeny of these long-living people had lower insulin, lower glucose, and they were more insulin sensitive, which was determined in various ways, including clump studies, which is the most valid and sensitive method for measuring, for assessment of insulin signaling. So in this study, at least, having inherited these longevity genes and being luckily in a cohort that has better life expectancy was significantly associated with improved insulin signaling. Next, please. And finally, last year, a study has appeared, which will be presented later in this meeting by Dr. Longo, which shows data from Laron dwarfs in Ecuador. Laron dwarfs, as I mentioned earlier in this talk, are people who have loss of function mutations in the gene coding for growth hormone receptor. So they are growth hormone resistant. They produce growth hormone. They don't respond to it. They are dwarfed. And here is, this data came from Ecuador, where Ecuadorian doctor, Dr. Guevara Aguirre, followed a large cohort of these patients for over 20 years. And they got lots of interesting clinical data on them. And shown here is one of his subjects. This gentleman is in his 60s, and his wife does not have this condition, so their children are normal. And you can see him here with his 5-year-old daughter, which is almost as tall as he is, and his boys who are 7 and 10, and you see they are already a lot taller than their father. So you can see it's an extreme phenotype in human also. And what was very interesting were some data on the causes of death and on the health problems of these people. Next. Summarized here is that out of approximately 100 people that were followed and looked at, nobody got diabetes. And consequently, of course, nobody died of diabetes. There was one incidence of cancer, which was successfully treated, and nobody died of cancer. And incidence of cancer and cancer death among relatives of these people and also in the general population of Ecuador is somewhere between 15 and 20%. And the incidence of diabetes is somewhere around 6 or 5%. So you can see that these people had rather remarkable degree of protection from both diabetes and cancer. And you also can see how this echoes findings in the Laron dwarf mouse, which gets cancer only half as often as a normal mouse and has improved insulin signaling. So next, please. Yet another question which invariably comes up when we think about, what this work means is, and this already was brought up in one of the talks earlier today, how can loss of normal function be good for anything? How can it make sense? And people interested in evolution, please go back, please. People interested in evolution of aging came up with a concept called antagonistic pleiotropy. And this concept implies that if genes are good for any traits that promote reproductive success, then they will be selected for and they will persist in the population, even if they have negative effects in the post-reproductive phase. And this will relate to the fact that in the natural situation, first of all, very few organisms become old. And second, that from the evolutionary perspective, longevity is not terribly relevant. And also, force of natural selection after the reproductive period becomes very weak or perhaps non-existent. So growth hormone, I think, fits very nicely with this concept because in a younger animal, it promotes growth, promotes sexual maturation, physical stamina. All of this means earlier acquisition of fertility. And in fact, fertility early in life with higher growth hormone levels tends to be better, even if growth hormone levels are pathologically increased, which can be done in mice by transgenes. In very young age, this has positive impact on reproduction. However, later on, the same signaling pathway, growth hormone signaling pathway, apparently accelerates the rate of aging, reduces stress resistance, reduces defense mechanisms, promotes cancer. So it causes these detrimental effects. And this perhaps explains why taking away somatotropic signaling results in extended lifespan. And of course, there are trade-offs. The trade-off would be delayed maturation and reduced fecundity. The Laron dwarf mice, both sexes are fertile. However, fertility quantitatively is reduced. Almost every parameter that we looked at in fertility is not as high as in a normal animal. So there are trade-offs. So next, please. This is simply to remind you and me that I'm just speaking here for many people. And these are my students, colleagues, fellows at our institutions listed on the left and other institutions on the right. And in the interest of time, I will not go over the individual contributions. And of course, this work couldn't be done without significant financial support. Doing longevity studies in mice, you're talking of 4-year experiments, so it costs money. And so we are very grateful to the agencies that supported us, starting with the National Institute on Aging. And since I think we have a few more minutes, I wanted to finish this talk with a joke. You know, when you work with aging, you run into lots of jokes about aging from colleagues and friends and magazines related to aging. And I think many of these jokes are terrible, and as I get older, they are less and less funny, but you know, they still are out there. But there is one that I thought you might perhaps enjoy, and also I am a zoologist by training, so it deals with animals, so it particularly appeals to me. So if I can ask for next slide. Next. And next, the question, And here's the answer. You're welcome.
Here comes New York, and that was a beautiful review. So you spend a lot of time in associating longevity with insulin sensitivity. And there will be a journal of gerontology that will come up in June, and I think it makes an important point. That you take lots of animals that the insulin signaling pathway was knocked out, it's IRS or other models. And on the other hand, insulin sensitivity was increased, for example, by increasing GLUT4 glucose transport. The relationship between insulin sensitivity and insulin resistance has nothing to do with longevity. As by the way, the nematodes and the flies are insulin resistant. So I think while I share the comment that in human mammalians, probably it's always better to be insulin sensitive, the question is if insulin resistance as a stress response is not something that when we need it, we need it, and it's important to know because we don't want to take it away.
Well, I think this data do not imply that to live long you have to be insulin sensitive. I think what this data tell us that in these animals with well-documented, consistent, and extreme extension of longevity, enhanced insulin signaling combined with low insulin levels, and I think that's very important. Combination of low insulin levels, enhanced insulin sensitivity is a good set of characteristics, and I think a strong candidate mechanism for aging. When aging is extended by other means, this relationship may not exist. And as you know, and as we discussed before, of course, in many cases, insulin resistance can be protective and can have its own benefits. Now, we are impressed with this partially because it fits our data from these animals, growth hormone resistant and growth hormone deficient animals. And also that there are this parallelism to clinical data, particularly data from the longevity, Leiden longevity study and similar studies. And also, I think it fits nicely with the results of anti-diabetic drugs that we just heard about this morning. But of course, I'm aware that there are mutant mice that are insulin resistant and long-lived. The first one that was described was the transgenic with the overexpression of work from Japan, I forget the gene. Anyway, in these transgenic animals, there's insulin resistance and extended life. And the authors of this study, in fact, suggested that increased longevity of these animals may be due to reduced insulin signal. That yes, they produce lots of insulin, but they are insulin resistant, and therefore the insulin signaling is reduced. So, you can interpret this data in various ways. And I did not want to leave you with the impression that insulin sensitivity explains everything, but I think it's an important factor in these animals that we're working with.
I enjoyed a lot your talk, as usual. I have a question for you. What about the thyroid function in this mouse? Because we were among the first many years ago to show that in centenarians the thyroid function was reduced. What about these mice? And related to this, do you have any data about the mitochondrial function?
thank you very much. These are extremely interesting questions. Well, the answer would be a little bit different for these two mutants, because in the Ames dwarf mouse, there is a primary deficiency of TSH, so of course they are severely hypothyroid. And Rich Miller showed that if you take the very similar dwarf mouse, the Snell dwarf mouse, and you give it water with thyroxine to drink through its whole life, it will live shorter. So the fact that they are hypothyroid perhaps is a contributing factor to their longevity. However, in the Laron dwarf mice, the growth hormone receptor knockout, there are only very minor changes in thyroid function, which are also a little different in females than in males, and both sexes are long-lived. So in these animals, probably the contribution of hypothyroidism is either minimal or perhaps not involved. I will have a chance later at these meetings, I think tomorrow morning, to talk about metabolic data in these animals, and which indirectly I think impacts mitochondrial function. They have, to give you just some highlights, they have consistently increased the expression of PGC1 alpha, so they presumably have enhanced biogenesis of mitochondria. There is evidence from Dr. Brown-Borg that they have reduced production of reactive oxygen species, so the mitochondria seem to function more effectively. And from indirect calorimetry work that Rehan Westbrook, a doctoral student in our lab, did, we know that they have reduced respiratory quotient, so they have enhanced utilization of fatty acids, and in fact they have also increased activity of enzymes involved in beta oxidation, which would also go with improved mitochondrial efficiency and reduced ROS production. So they have a shift in mitochondrial function. They are surprisingly not hypometabolic, and these are the data I'll be showing tomorrow. They have in fact increased oxygen consumption per gram body weight, and also increased oxygen consumption per gram of lean body mass. So they have increased conversion, increased oxygen consumption, increased conversion, which, you know, I don't want to give tomorrow's talk here, but I think it importantly relates to thermogenesis. They are small, they lose heat at different rate, but they have reduced RQ, and I think that this enhanced utilization of fat as fuel, and in this way improved mitochondrial function, is a very important part of the phenotype and another very strong candidate mechanism for extended longevity. As you remember from one of the early slides, you know, there's a long list of candidate mechanisms, you know, and this enhanced insulin sensitivity is just something that we spend lots of time working on, but there are many others.
Sean Kerman, University of Southern California, in the back.
Mm-hmm.
I was wondering, so one potential difference is that C. elegans can inhibit insulin IGF-1 signaling at any point during life and get the lifespan extension phenotype. Is there a window in development where you can supplement the Ames dwarf mice with growth hormone, suppress the growth defect, and still maintain longevity?
We are working on it right now, and we're particularly interested in replacement with growth hormone early in life, so we can get them to grow and to become more phenotypically normal and seeing what would happen. And we have done a number of these experiments, and where we got a large change in body size and other expected changes on growth hormone administration, and we got no impact on longevity. However, we had one experiment which we reported a couple of years ago, where we increased the intensity of growth hormone treatment by injecting it twice daily at fairly high dose. We also started early in life. We started at 2 weeks of age. And this is very early in development. These animals are developing a little slower than normals, so 2 weeks of age is very young. And under these circumstances, 6 weeks of growth hormone treatment significantly shortened their life. So apparently there is some, I don't know, programming is the word that many people object to, but there are some early effects, perhaps epigenetic, that determine life expectancy. This was one experiment, but the results were very striking. And we are working on it now. We have other experiments in progress.
Of course, I'm struck by the many parallels between the Ames dwarf and dwarf and the very volatile dwarfs that we studied. You pointed out the higher fat content, which is similar, the lower respiratory quotient, and the actual increase in metabolic rate, which is a little paradoxical. Where does the caloric restriction plus dwarfism, the Ames plus caloric restriction, which is the longest animal we looked at, where does that fit into this? Does it also somehow still have high metabolic rate, even higher metabolic rate than lower respiratory quotient?
Well, the differences in oxygen consumption are evident in caloric-restricted animals also. Just the nature of this work makes it very difficult to get convincing answer to the question whether the effect is bigger or not. We can show that it is significant at 2 different levels of calorie restriction, and it is significant at ad libitum feeding. But, you know, these are separate experiments done at different times, somewhat different age, you know. So we really don't have data that would allow meaningful comparison when the effect is stronger. Now, the overlap with calorie restriction from looking at gene expression data and looking at life extension and some of the insulin signaling parameters, it seems that the growth hormone receptor knockout is more phenotypically similar to caloric-restricted animal, and the dwarf is more different. And maybe it's because of hypothyroidism, you know, which I was just discussing. Maybe it's something in development. You know, the dwarfs do have receptors for growth hormone and for other hormones from that family, so they can respond to the placental hormones and to maternal hormones, at least in the first week of life or so, through milk. So maybe that makes them a little different, I don't know.